How to prove diffusion for lazy people created by lazy students. Let's draw up the concept of the experiment. Put two tied-up bags of iodine and a starch solution in two beakers filled with the starch solution and iodine. The iodine solution is a hypotonic solution, and the iodine will flow into the bag of starch, turning it blue. The starch solution is a hypertonic solution, and the iodine will flow out of the bag and into the solution, turning the outside solution blue. This simulates the scientific principles of cytolysis and plasmolysis. The starch cell in iodine simulates cytolysis because since the starch cell is in a hypotonic solution, the iodine will flow into the starch cell until the water concentrations balance out, reaching homeostasis. As a result, the cell may explode. The iodine cell in starch simulates plasmolysis because since the iodine cell is in a hypertonic solution, the iodine will flow out the cell and into the starch solution until the percentages of water concentration even out, or until it reaches homeostasis. As a result, the cell may shrink and die. Another crucial piece of information that people often forget when doing this is that the dialysis tubing used to simulate the cell membrane is semi-permeable like the cell membrane, allowing the flow of liquid but not particles. As a result, iodine flows out in a hypertonic solution, not the starch flowing in, so the solution in the beaker should turn blue, not the solution in the cell. This information is crucial because when analyzing plant cells in a lab, one will realize how it is just like the simulations. Now, get two beakers, one with 2 tablespoons of starch, a graduated cylinder, 2 binder clips, a 1-foot strip of dialysis tubing, string, and iodine. Create the starch solution by mixing 200 milliliters of water into the starch. Now create the iodine solution by mixing the 30 drops of iodine with 200 milliliters of water. Now to create the cells, cut the dialysis tubing in half. Tie one of the ends of the piece of dialysis tubing and fill the other end with 10 milliliters of starch solution. Tie the other end and clip a binder clip on it. Great, now we have one cell. Tie one end of the other piece of dialysis tubing and fill it with 10 milliliters of the iodine solution. Great, now we have the other cell. Submerge the starch cell into the iodine solution and the iodine solution into the starch solution. Now observe for 15 minutes. Great, the starch cell should turn blue since it was submerged in a hypertonic solution. The iodine will flow into the starch cell. The iodine cell will shrink, because it is submerged in a hypertonic solution, and the iodine will flow out of the cell. The starch-filled beaker should turn blue given enough time, and the iodine-filled beaker should have decreased in amount. Well, now you know how to prove that diffusion exists. Diffusion, the process of moving a particle from a region of high concentration to a low concentration, can be proven. So whenever you're in a heated debate with a flat earther who believes the moon landings are fake and the earth is flat, well, at least you can convince him that diffusion is real. Or maybe not. Goodbye.